Well, hey now, living environment fans. As you noticed, once again, I'm out of the office today. Why is it always Thursdays? I don't know. That's when these things are planned, but don't worry, I'll be back tomorrow. But today, you have plenty of stuff to do, including this Bellringer. And of course, we've been studying DNA, the double helix of the deoxyribonucleic acid molecule. And we've been studying how it copies itself and how cells copy themselves, and then how we use DNA for identification purposes. We're going to move on and we're going to see what does DNA actually tell the cell to do. All right, let's do the Bellringer. All right, pop quiz. In eukaryotic cells, where is most DNA found? Very good. It's in the nucleus, right? In eukaryotic cells, DNA in the nucleus is usually long strands, but during mitosis, they coil up into good. They're called chromosomes then. All right. And of course, the DNA double helix, the ladder, the rungs of the ladder are made up of nitrogen base pairs. And of course, base pairs always match the same way. A matches with T, G matches with, that's right, C. All right. So you got DNA floating around in the nucleus, right? And if it's not going through mitosis, it's not copying itself, then the DNA is doing what it's supposed to do. It's telling the cell how to proceed. And what DNA tells the cell how to do is how to make proteins, including enzymes, right? Including the enzymes that run the making of proteins. All right, let's look at inside the nucleus. A segment of DNA that codes for a protein, we call a gene. Right? Not all DNA codes for proteins. Some of the DNA we call junk DNA, although it has other jobs. But the segment of DNA that codes for a protein is called a gene. And in human cells, you've got about 30,000 genes. Right? One typical cell, though, however, will probably only read about 2,000 of the 30,000 genes. All right, and that's how cells differentiate, how you get some cells to be toenail cells, some cells to be liver cells. Right? They don't read the same genes. We'll talk about how they know what genes to read in a minute, but when they read the genes, it's almost like replication. You got this long strand. Well, to read the gene, you have to separate. You've got to use that helicase enzyme. But this time, instead of DNA polymerase, you use another enzyme called RNA polymerase. And RNA polymerase attaches onto the strand of DNA after it's been split, and it reads one side from the 3 to the 5 side, reads that side of the DNA strand, and it matches up C's with G's and T's with A's and A's with U. Yeah, that's weird. mRNA or RNA has U in it. Isn't that weird? We'll look at that in a second.
All right. Hey now. Mr. Kovacs class. It's Mr. Kovacs class. You might learn a ton. It's Mr. Kovacs class.
He's interesting too.
It's Mr. Kovacs class. And he's super cool. It's Mr. Kovacs class.
So to read a gene, right, RNA polymerase has to match, let's see, it matches A's to the T's, G's to the C's, C's to the G's, but it matches U's to the A's. Right? It's weird. There's no T in RNA. It's only got U. I like to think of it, DNA doesn't care about you, but RNA cares about you for sure. And then what happens is you get this strand of, it's kind of like one side, a copy of one side of the DNA molecule. That strand is called messenger RNA. And this whole process here is called transcription. Transcription. It happens in the nucleus. Transcription. It's the copying of a gene from DNA to an mRNA template. Transcription, copying a gene from DNA, right? The gene's coded in DNA. It's copied to an mRNA template in the nucleus. So the next step is, well, you've got to turn that gene into a protein. You got to take this mRNA and move it out of the nucleus into the cytoplasm to an object that can make proteins, which we know are called. Nice, you remember that cell part was called the ribosome. Very good, very good. All right, so this is transcription. The first step for cells knowing what to do is to transcribe genes. They do it in the nucleus, right? They do it usually during G1 phase or the G2 phase, right? Usually while they're in the replication phase, they're too busy replicating to do a lot of transcription. And when they're in mitosis, they definitely don't do any transcription. All right? So today you've got the triple header of things to do, of course. After this Bellringer, you've got two other things that you need to do. I've got an Edpuzzle video about gel electrophoresis for you to take a look at. And then I put some notes in your classroom about DNA fingerprinting that I want you to complete. And then if you have lab today, you're going to do the DNA fingerprinting lab, which is pretty self-explanatory. There's three things for you to do. You open up the mystery of who licked the lollipop, and you have to solve that mystery. And then you're going to do a simulation of using a gel electrophoresis machine. And then the third thing you do in that lab, of course, is you solve some crime. Well, you solve some DNA fingerprint problems, like who is the father, who's the suspect, those kinds of things. And then you don't have to do number 8. You don't have to do number 8. Remind me to tell you not to do number 8. All right, I'll see you tomorrow.